Mitochondria. Albert von Kolliker in 1857 defined mitochondria as a thread-like or granular cytoplasmic organelle. Mitochondria is also termed as powerhouse of the cell, chondrosome, and sarcosome. Is mitochondria a cell organelle? Yes, mitochondria are spherical to oval-shaped organelle found in all eukaryotic cells. Mitochondria produces energy as ATP through a series of chemical reactions. Mitochondria are membrane-bound cell organelles that generate energy which are needed to power the cell's biochemical reactions. Hence, it is called as powerhouse of the cell. Can a cell live without mitochondria? A cell cannot survive without mitochondria, the organelle which boost the human cells. Without mitochondria, higher animals would not exist because cells obtain energy from mitochondrial ATP synthesis. Mitochondria enable cells to produce 15 times more ATP, which helps in the cellular activities. How can we compare the mitochondria with a human life? The mitochondria are like a battery of a system. Mitochondria of the cell make energy for the cellular respiration of a cell as like battery which provides energy to the system. If the mitochondria are damaged or a breakdown occurs, the body cannot properly recharge and hence produce energy. Next, we will learn about the interesting facts of mitochondria. Mitochondria can quickly change its shape and move around the cell. When the cells need more energy, the mitochondria can reproduce by growing larger and then dividing. The number of mitochondria in a cell varies among the eukaryote forms depending on the energy requirement. Cells that require more energy to complete their metabolism will have a high number of mitochondria. Human muscle cells are the only cells with more number of mitochondria. Do you know how mitochondria has originated and evolved? Mitochondria arose through endosymbiosis more than 1.45 billion years ago. Margulis in 1970 suggested that mitochondria evolved from free-living bacteria through symbiosis within a eukaryotic host cell. Symbiotic origin. Symbiosis is defined as the interaction between two different organisms living in close physical association, typically to the advantage of both. Altman and Sniffer in 1890 suggested that the mitochondria might have originated from prokaryotic cells like bacteria. The bacteria entered the cell as parasites. In the course of time, bacteria maintained a symbiotic relationship with the eukaryotic cells and thus formed the mitochondrion. Who discovered mitochondria? Mitochondria were first observed by Albert von Kolliker in 1857, a physiologist, and later coined as bioblast by Richard Altman in 1886. Carl Benda in 1898 renamed it as mitochondria. The first recorded information of mitochondria in plant cells was by Friedrich Maves in 1904. Role of mitochondria in prokaryotic cell. Prokaryotic cell lack mitochondria and instead produce their ATP on their cell surface membrane. Prokaryotes do not have mitochondria for energy production. Prokaryotes generally use electron transport chain in their plasma membranes to provide much of their energy. Role of mitochondria in eukaryotic cell. Mitochondria are spherical to elongated oval-shaped organelles that can be considered as the power generators of the cell. The number of mitochondria present in a cell depends upon the metabolic requirements of a cell and may range from a single large mitochondrion to thousands of mitochondria. Occurrence of mitochondria in eukaryotic cell. Mitochondria are found in the cells of eukaryotic organisms including plants and animals. Cells that require a lot of energy such as muscle cell can contain hundreds to thousands of mitochondria. Mitochondria entirely lacks in human red blood cells. Location of mitochondria in eukaryotes. Mitochondria are located in the cytoplasm of a cell along with other organelles. Mitochondria are found in all human cells except in red blood cells, that is RBC. There are usually many mitochondria found in a cell depending upon the function of the cell. Size and shape in eukaryotic cell. Mitochondria are organelles typically that range in size from 1 to 10 micrometer in length found in the cytoplasm of eukaryotic cells. Mitochondria contain inner and outer membranes. The outer membrane is 60 to 70 angstrom thick. Mitochondria are typically spherical to oval in shape, and they vary in shape from bean-like units to a lattice-like form. Mitochondrial DNA. Mitochondria contain one or more DNA called mitochondrial DNA, that is mtDNA. The mitochondrial DNA is circular in shape and can self-replicate. The mitochondrial DNA is associated with cytoplasmic inheritance. The mitochondrial rRNAs are encoded by mitochondrial DNA. The ribosomal proteins are encoded in the nuclear genome, which are synthesized on cytoplasmic ribosomes and then are imported into the mitochondria. Chemical nature of mitochondria. Mitochondria are composed of lipids and proteins. The mitochondrial matrix contains a large number of enzymes, coenzymes, organic and inorganic salts, DNA, RNA, and ribosomes. Mitochondria contain 65 to 75% of protein, 25 to 30% of lipid, 0.5% of RNA and DNA. The lipid part of mitochondria is composed of 90% of phospholipids, 5% of cholesterol, and 5% of fatty acids. Structural features of mitochondria in eukaryotes. Mitochondria is composed of the outer membrane, the inner membrane, the intermembrane space, the cristae, and the matrix. Outer membrane. The mitochondrial outer membrane is a double phospholipid membrane. Porins present in the outer membrane of mitochondria are channels formed by the proteins in the phospholipid bilayer. Porin channels allow smaller proteins to enter freely by diffusion. The outer membrane is associated with the smooth endoplasmic reticulum. Inner membrane. The mitochondrial inner membrane is made up of a phospholipid bilayer which are embedded by proteins that serve to carry out the electron transport chain. The inner membrane has folds called cristae that increase its surface area. The space within the inner membrane of a mitochondrion is known as the matrix. Intermembrane space. The intermembrane space, otherwise known as IMS, of mitochondria is enclosed by the outer and the inner membrane of the organelle. The IMS can be subdivided into two distinct subcompartments, the intracristae space and the lumen between the outer and the inner membrane, which is also called the external IMS. Both subcompartments are separated by cristae junctions. Cristae. Cristae are the finger-like invaginations of inner mitochondrial membrane into the matrix. Cristae develop as in pushing projection into the central space from the inner membrane. The cristae are covered with small particles called elementary particles or F1 particles. Each F1 particle has a base, a stalk, and a head. Matrix. The inner chamber is filled with mitochondrial matrix, is a gel-like material. The matrix contains 70 S ribosomes and mitochondrial DNA. The matrix contains the mitochondrial genome and the enzymes of the tricarboxylic acid, which is otherwise called as TCA cycle. Functions of mitochondria. The main function of the mitochondria is to produce energy for the synthesis of ATP, Mitochondria help the cells to maintain proper concentration of calcium ions within the compartments of the cell. It can regulate activation, differentiation, transcription, and survival of immune cells, and it releases signal to regulate transcription of immune cells. The mitochondria plays an important role in the process of apoptosis or programmed cell death. It helps in building certain parts of blood and hormones like testosterone and estrogen. The mitochondria in liver cells have enzymes to detoxify ammonia. Mitochondria plays a major role in cellular respiration. Cellular respiration. Mitochondria are the respiratory centers of the cell. They bring out oxidation of various foodstuffs. During oxidation process, the foodstuffs are degraded to carbon dioxide and water with the release of energy as ATP. Cellular respiration involves the following steps: glycolysis, oxidative decarboxylation, Krebs cycle, electron transport chain, and oxidative phosphorylation. Glycolysis. Glycolysis is also known as the Embden-Meyhoff pathway. It is the process by which one molecule of glucose is converted into two molecules of pyruvate, two hydrogen ions, and two molecules of water. Pyruvate molecules then proceed to the link reaction where acetyl-CoAmine is produced. Acetyl-CoAmine then proceeds to the tricarboxylic acid cycle. Glycolysis takes place in the cytoplasm but not in the mitochondria. Oxidative decarboxylation. The pyruvic acid produced during glycolysis enters the mitochondria. With the help of the enzyme pyruvic acid dehydrogenase, pyruvic acid undergoes decarboxylation and oxidation to acetyl-CoAmine. During the process, two hydrogen atoms are released and combined with NAD and is reduced to NADH2. NADH2 enters the electron transport system. Krebs cycle. It is the oxidation of pyruvic acid into carbon dioxide and water. It was first described by Krebs in 1936. Krebs cycle is also known as citric acid cycle, tricarboxylic acid cycle. It occurs only in the presence of oxygen, hence it is an anaerobic process. In Krebs cycle, two molecules of carbon dioxide are released and oxaloacetate is recycled. Energy is stored in ATP and other high energy compounds like NADH and FADH2. The process of Krebs cycle. Formation of citric acid. The acetyl-CoAmine combines with 4-carbon compound oxaloacetate to form 6C-citrate catalyzed by citrate synthase. Dehydration. Citrate is converted to isocitrate by enzyme conitase. Dehydrogenation. Isocitrate undergoes dehydrogenation and decarboxylation to form 5C-ketoglutarate by isocitrate dehydrogenase. The release of carbon dioxide and NAD+ is converted to NADH. Oxidative decarboxylation. Ketoglutarate undergoes oxidative decarboxylation to form succinyl-CoAmine, a 4C compound by ketoglutarate dehydrogenase enzyme. One molecule of carbon dioxide is released and NAD+ is converted to NADH. Succinyl-CoAmine forms succinate. The enzyme succinyl-CoAmine synthetase catalyzes the reaction and ADP forming ATP. Oxidation. Succinate is oxidized by the enzyme succinate dehydrogenase to fumarate and FAD is converted to FADH2. Hydration. Fumarate gets converted to malate by addition of one water molecule by enzyme fumarase. Dehydrogenation. Malate is dehydrogenated to form oxaloacetate by malate dehydrogenase and NAD+ is converted into NADH, which combines with another molecule of acetyl-CoAmine and starts the Krebs cycle. Electron transport system, which is otherwise called as ETS, is of enzymes and coenzymes where the reduced coenzymes like FADH and NADH are oxidized to release energy. The energy released in ETS is used for the synthesis of ATP. The synthesis of ATP is called as oxidative phosphorylation. The steps involved in ETS are the removal of hydrogen from the substrate NADH or FADH by the enzyme dehydrogenase. The hydrogen atom becomes ionized into protons and electrons. The hydrogen ion reduces the coenzyme NAD. The NADH is oxidized into NAD by transferring its hydrogen ion to FAD, which acts as the hydrogen carrier. From FAD, each hydrogen ion is discharged in the cell fluid and electrons are passed on to the cytochrome B, C, A and A3. From the cytochrome, the electrons are given to the enzyme cytochrome oxidase. The cytochrome oxidase finally discharges electrons to oxygen. This oxygen unites with hydrogen ions, forming water. Oxidative phosphorylation. The process of ATP formation which occurs during the oxidative reaction of Krebs cycle is known as oxidative phosphorylation. With the release of ATP, energy carriers, they are converted back to ADP. Disease caused due to defects of mitochondria in human. Kearns-Sayre syndrome, otherwise known as KSS. Stroke-like episodes, which is otherwise known as MELAS, MELAS. Myoclonic epilepsy with raised red fibers, which is otherwise known as MERF, MERRF. Leigh syndrome, otherwise known as LS. Neurogenic weakness with ataxia. And retinitis pigmentosa, which is otherwise known as NARP, NARP.